In the series of short videos, this small presentation is on RNA polymerases. This is the usual disclaimer. Now, these enzyme RNA polymerases, whether they are present in eukaryotes or prokaryotes, what do? They catalyze the they catalyze the synthesis of RNA from DNA. That is, they mediate the process of transcription. The one which is present in prokaryotes, the bacterial RNA polymerase, is a large, approximately 500 kilodalton molecular weight enzyme made up of multiple subunits. It's a multi-subunit assembly, and as many as 4 different types of subunits are present in it. The subunit composition and the functions of individual subunits are shown in this particular table. For example, there is alpha subunit. There are 2 of them present. There are 2 alpha subunits present, each one having a molecular mass of 37 kilodalton. And what is their role? What is their function? What is their job? Their job is to bind to regulatory sequences present on the DNA. Similarly, there is beta subunit. There is one present, and the molecular mass is 151 kilodalton. And the active center, the catalytic center of the enzyme is located on this. It is responsible for catalyzing the formation of the phosphodiester bonds between 2 ribonucleotides. There is one beta prime subunit. The molecular mass is 157 kilodalton. And what does it do? It binds to template DNA. It helps RNA polymerase bind to the template DNA. And there is sigma subunit. Okay, the one which is present in standard enzyme has got a molecular mass of 70 kilodalton. Please remember that there are multiple sigma subunits present, each one with a different molecular mass and each one with a different job of identifying, recognizing the promoters of a given set of genes. The job of sigma subunit, irrespective of its molecular mass, is what? Is to help RNA polymerase recognize the promoter, the transcription initiation sites, and initiate transcription. So irrespective of molecular mass, the job of sigma subunit is to help RNA polymerase identify the transcription initiation sites. We call them as promoter. So for example, this is the holoenzyme, the one which is made up of all the 4 different types of subunit. There is one sigma, there are 2 alpha, there is one beta, and there is one beta prime. Okay, this sigma subunit, after its job is over, after it has helped the RNA polymerase, the enzyme, recognize the promoter, it leaves the complex. And then what remains is called as the enzyme minus the sigma subunit is called as core enzyme. And the rest of the transcription is carried out by this core enzyme. So bacterial RNA polymerase, what are its functions? Its functions are that it searches the DNA for initiation sites, the promoters. Okay, it is the responsibility of this enzyme to identify, to recognize the promoter. And a small genome, the one which is present in E. coli, may have as many as 2,000 promoters. See, the genome size of E. coli is 4 into 10 to the power 6. Okay, so it is almost 1,000 times less than that of the human genome. But such a small genome also has close to 2,000 promoters. So it is the responsibility of RNA polymerase to identify the promoter. What does it do next? It unwinds a short stretch of DNA to generate a single-stranded DNA, so that a template is made available, because it takes instructions from that particular DNA template to synthesize the RNA molecule. Okay, so it is again the responsibility of RNA polymerase to unwind a small short stretch of DNA so that a single-stranded template is made available for it to take instructions from. Then it selects the correct ribonucleotide from among the pool of ribonucleotides. Okay, based on what is there on the template DNA, the complementary ribonucleotide is picked up from the pool of ribonucleotides, and it is again the responsibility of this enzyme to catalyze the formation of phosphodiester bond between the 2 ribonucleotides. It also detects the termination signals where the transcription process terminates, and it interacts with the activator and repressor proteins so that the transcription can be regulated. The rate of transcription can be regulated by interacting with the activator molecules and repressor molecules. If it interacts with an activator molecule, obviously the rate of transcription will increase. And if it interacts with the repressor molecule, the rate of transcription will decrease. Contrary to prokaryotes, in eukaryotes, you have three different types of RNA polymerases. You have three different types of RNA polymerases in higher organisms. Their location is also different, and their work profile, their responsibility also differs. For example, you have RNA polymerase 1. Where it is present, it is located in nucleolus, a small organelle present within the nucleus. And what does it do? It is responsible for the synthesis of these ribosomal RNAs. 18S, 5.8S, and 28S ribosomal RNA are synthesized by RNA pol 1. RNA pol 2, it is the main RNA polymerase present in eukaryote. It is present in nucleoplasm, and it is responsible for the synthesis of messenger RNA and small nuclear RNAs, snRNAs. Messenger RNA precursors, we call them, because messenger RNA in eukaryotes undergo post-transcriptional modifications. And there is one more RNA polymerase 3. It is also present in nucleoplasm, but it is responsible for the synthesis of all different types of tRNAs and 5S rRNA, 5S ribosomal RNA. It should be 5S ribosomal RNA. Okay, and these three RNA polymerases can be distinguished from one another because of their response to what is called as an alpha-amanitin. Alpha-amanitin is a toxin which is present in a poisonous mushroom called Amanita phalloides. Okay, so RNA pol 1 is found to be insensitive to this toxin, whereas RNA pol 2, contrary to RNA pol 1, is found to be highly sensitive to this alpha-amanitin. And RNA pol 3 requires a relatively higher concentration of this toxin to get affected. That means it is moderately sensitive. Of these three RNA polymerases, RNA pol 2 is analogous to the prokaryotic RNA polymerases. Means it is the main RNA polymerase which is involved in the synthesis of messenger RNA. Thank you.